Hello, my name is Laura Saavedra and I'm a researcher from CONICET in Argentina. I would like to share with you the results from our group about the roles of autophagy in the Mosquito Comitrella patens. Briefly, we found that the protective role of autophagy under nutrient deficiency is conserving bee patens, but also provide evidences of this mechanism to sustain the apical growth of protonemata during darkness under optimal growth conditions. So, autophagy is a highly conserved catabolic process in eukaryotic cells triggered by environmental or internal cues in which cytoplasmic components are sequestered into double membrane structures called autophagosomes and then are delivered to the backbone for digestion and recycling. Autophagy is virtually involved in all aspects of plant physiology. Most knowledge and major contributions in the field of plant autophagy arise from studies in seed plants. However, in the last years, availability of newly sequenced genomes from algae and bryophytes allow us to dive into evolutionary aspects of plant autophagy as well as species-specific roles of this mechanism. In particular, we are focused on bryophytes and are studying using the most Fiscomitrum patens as a model system. P patens is also a model system for apical growth because this filamentous tissue, composed of two different types of cells, chloronema and caulonema, grows by apical extensions. These cells exhibit different characteristics. So while chloronema cells are rich in chloroplast and perform a photosynthetic function and have slow growth rate, caulonemata cells are poor in chloroplast, have a function more related to substrate colonization and nutrient acquisition, and have a faster growth rate. So the questions we want to address are, first, are autophagy, core machinery, and responses to nutrient deficiency conserving P patens? And second, does autophagy have a role in apical extension and or differentially regulate the nutritional state of these cells? So we first searched for autophagy ortholog genes in the P patens genome and found that the core ATG machinery is present. Then we select ATG5 and ATG7 genes and generate local knockout lines and perform nitrogen and carbon starvation treatments considered as canonical methods to induce autophagy in plants. So what we can observe in the figure and in the graph of chlorophyll fluorescence, ATG5 and ATG7 lines were hypersensitive to carbon and nitrogen starvation. So here we have the treatments with light without nitrogen, darkness, or darkness and sucrose. These results support a role of autophagy under nutrient deficiency. In addition, we noticed that even the mutants under optimal growth conditions start dying earlier, as you can see here by the yellowish color in the center of the colony. Next, we analyze the protonemata of these mutant cells under optimal growth conditions, and we could observe a high number of dead cells in the 7-day-old tissue in comparison to the wild type, pointing out that autophagy has an important role in moss. Then we measured the lengths of protonema subapical cells, of chloronema cells, and caulonema cells, and found that both types of cells have shorter cell lengths compared to the wild type. In addition, we measured the growth rate of also the growth rate of chloronema and caulonema cells, and whereas in chloronema cells we didn't found significant differences, in caulonema cells we found that ATG7 mutants grow slower than the wild type. These observations lead us to analyze if autophagy was induced in protonemata under these conditions. So we measured the expression of the autophagy marker gene ATG8 after 16 hours of light period, which is our control, and then we compare it with sample of 2, 4, 8, and 25 and 24 hours of darkness, or darkness supplemented with sucrose. The 6 pp80 genes were upregulated at night at 2 with a peak at 4 hours and then diminished at 8 hours, or were highly induced in the case of extended darkness with or without sucrose, showing that autophagy is induced in darkness. So we next analyze autophagy induction, monitoring the presence of autophagic vesicles in this tissue using a fluorescent line, which is a GFP ATGB line under the control of its endogenous promoter. We found that in the apical chloranemata cells, autophagic vesicles accumulate significantly at 8 hours of darkness in comparison to light conditions or darkness of subapical cells. However, in caulonemata, the accumulation of autophagic vesicles was already at 4 and at 8 hours, indicating an early induction in these cell types and thus a differential response of these cells. To conclude, the data presented here show that the protective role of autophagy under extended carbon and nitrogen starvation is conserved in Comitrella patens. In addition, our data provides a relationship between autophagy and apical growth, suggesting that autophagy could sustain the growth of protonemata cells, in particular that of caulonemata, which exhibit a higher growth rate. Finally, I want to introduce our research group led by Professor Ramiro Lascano, and I want to thank these institutions for financial support, and thank you for your attention.